So I'm very honored to be here today and wanted to also celebrate with you together about 140th anniversary plus grand opening of these quantum initiatives. So I'm super excited, and also I've been more excited as I can have this opportunity to share our recent works on the quantum. And I'm working for the university and Cambridge University, and I'm involved in a number of different departments as my work has been between biology, medicine, and computer science. So I hope to convince you also how and why we need quantum on drug discovery. So first of all, previous two speakers give fantastic kind of introduction and background in both quantum and biomedical side, but I wanted to just reiterate. So basically, everyone knows about quantum is fast and also it's very powerful, so there is no doubt. But our kind of my lab's more interested in whether quantum really provide kind of truly novel insight than AI or classical algorithms. So we are more focused on this side to see whether this is true or not. Also, we wanted to kind of validate this hypothesis scientifically. So why then I'm working on drug discovery and wanted to apply quantum on drug discovery? First of all, you are seeing just one pill of drug and one rocket that may send us to the moon or even Mars. So it's an obvious question, but I think which kind of project is more expensive than the other? Yeah, it's an obvious question because I'm talking about drug discovery. Of course, drug discovery is more expensive, like even 4 times, usually 4 times more expensive than just developing a rocket. So this is really, really expensive project. And also, it takes so long time, more than 10 years easily. If it's finished in 10 years, you are lucky. And obviously, as I said, it needs a lot of money. But more problem is very, very complicated stage, and we need quite a lot of people, not just scientists like me or researchers, but also clinicians, surgeons, and even more importantly, we need patients and their family. It's not just like a science program. It's a really, really societal kind of effect. But finally, the biggest issue in this program or project is like it has very, very low success rate. So it's only now, there is a kind of number here, somewhere 12%, but that's outdated. Most recent one from Nature Drug Discovery saying is around 8%. And in cancer, as Professor Zhang just said, ironically, cancer is one of the areas that a lot of research is going on. However, the success rate, or average rate, is just 5%. So this is a really daunting challenge. So we need some kind of breakthrough. And since 2017, there are a lot of kind of conversation or discussion or even kind of debating is going on, has gone. And now I think everyone knows AI is really needed for drug discovery. And due to the time limit, I can't explain everything about AI drug discovery, but if you are interested, please read this nice paper published by Wellcome Trust and Boston Consulting Group in 23. It's publicly available, and then you can just really read it, and it's a really nice document. I only take 3 shots from this nice paper, one of which is, again, highlight the key pain points in early drug discovery. So we are academic, and actually we can't really working on clinical trials, but we are more working on early drug discovery stage, where we can really find biological target, understand the disease mechanisms, and then we can actually develop chemicals or antibodies that actually treat our bodies. In here, there are a lot of pain points, unfortunately, and my lab is particularly focused on much, much earlier stage, like so-called target identification. And then also this report is nicely summarized what kind of AI activity is going on to tackle those key pain points. And there are quite a lot. One of which is like this middle part. I will come back to this in next slide. But also, this is what my lab is do quite a lot every day, which is a drug repositioning, but also analyzing multiomics data, which just Professor Zhang explained, and to understand the disease mechanisms and identify targets. So final slide of this is about then AI is really making an impact on drug discovery or not. And by stage, they highlight those two areas, which call hit to lead and lead optimization. This is where AI being used to predict the chemical compound structure, or as you know, AlphaFold, predict three-dimensional structure protein and find the binding site for the drugs. So this is really, really advanced, and I agree. A lot of AI has been used, and there is a lot of progress. So that means we now have a lot of kind of time saving and cost saving, which is great. As I show, it's 4 times more expensive than just a rocket project. So if we can come down by, I don't know, by half or even further, that's fantastic. Also, we can just do it not in 12 or 10 years, but just like do it in 3 to 5 years, which is also great. However, I think AI can do much better, which is we need more impact on the target ID and validation side. That's why my lab is working on this particular space. And in just one slide, I wanted to give very quick brief AI drug discovery work in my lab, and then moving on to the quantum side, because we need this slide to understand why we are doing this. So we are doing biomedical data-driven AI drug discovery. And in the first stage, also, again, Professor Jung really gave really nice background information, but we actually have quite a lot of different multimodal data sets. However, they are kind of scattered. Some are available through the collaboration, like in private connection, but also some are only available in public domain. Yes, there are a lot of data. The problem is how we can gather them and then actually make them integrated and harmonized. This is really a fundamental issue. As everyone say big data, but big data is of course, however, it's not just about the quality, but it's also about the quantity. Quantity, but it's not also about quality. So we really need high quality, high quantity data sets. So we spend quite a lot of time to prepare this. I think in the quantum situation is more or less similar kind of challenges. And then second stage, once we have this harmonized database, we are moving up to understand the multi-omics disease associations. That means we wanted to understand, for example, we are studying in lung cancer, we wanted to understand what type of genes causing the lung cancer. So we wanted to compare the lung cancer patient genomic, transcriptomic, epigenomic, whatever multi-omics data versus the non-disease healthy individuals. So in this stage, we identify so-called disease signature. This is somewhat like 100, let's say top 100 genes that are highly expressed in the only lung cancer patient than non-disease kind of individuals. Then we are very excited, like we already know, oh, they are 100, like really exciting genes that causing lung cancer maybe. However, the next question is obviously, then among those 100, are there any kind of small group that are working together, or are there any master regulator that controls most of those top 100 genes? In this typical and very simple one-dimensional data structure, there is very, very limited algorithm is available to apply on this. However, if we can put it into a little bit complicated data structure, like at least two-dimensional, like this called like a graph-based format. So we have this like a two-dimensional structure, and imagine this is just for the lung tissue, but if we have this is for the liver tissue, brain tissue, and other part of the body, then it could be stacked up to each other, and then that could be multi-layer, means it's going to be three-dimensional structure. So we can actually make this more sophisticated and complex data structure, which is one step closer to the real biology. And then we can run number of very exciting kind of algorithms, like machine learning, of course, but also network simulation or network algorithms. So in this stage, all the focus is to identify druggable pathway. So we just wanted to understand, as I said, not just name of genes, top 100, but we wanted to understand their connection, their relationship, and are there any kind of upstream genes that causing the real disease, and we need to target those specific genes also. So this is very interesting step, and then next two slides, I will jump onto this a little bit more. And then finally, what we identified is more or less two kind of lists. One is the really important biological target in this druggable pathway, that usually gene or proteins, and then also we can run the drug simulation on this network to identify the small molecule, like chemical compounds that maybe really nicely inhibit those biological targets. And we are finishing here as a computational work. However, this is not really the end of the project. In any type of project we're having in the lab, it's 100% collaboration. In fact, I also have collaboration with Professor Jung, but also I have quite a lot of collaboration with mostly clinician scientists, and the big pharma companies. Firstly, we can have the real data from them. Secondly, they are thankfully very willing to test or validate our predictions on their bench. That means we are going back to the wet lab and ask them to test our predictions, whether this is working or not, on the cell line, organoids, or mouse model, or any other animal models. So this is the true validation of our prediction. And then the really important part is this is not really the end of the story, but we are coming back to the first step. So that means we are not just finishing it one-off, but we are doing it iteratively so that we can increase the accuracy, specificity, sensitivity in the second, third, fourth round until we are satisfied. So this is very important steps, and this is what we usually do. And as I said, you showed just like a lovely schematic diagram of proteins kind of network, but in the reality, we are dealing with more than 10, 20,000 protein network. As you can see, it's really somehow lovely, but somehow challenging to identify important nodes in this massive network. That's why we need so-called, to identify key pathways, we need network algorithms. There are a number of different algorithms, but I wanted to particularly talking about random walk with the restart, which is invented by genius Alan Turing in Cambridge in 1950s, and this is often used still. So one of the examples is here. Here is only 164 proteins, and the lines are the interaction between 164 proteins, and probably you can spend some time with a cup of coffee or tea, and then by your naked eye, you may find some key important proteins in this complex network. However, as I said, we have a lovely tool called random walk, coined by Alan Turing. And this is basically in a couple of sentences. What it does is like after the calculation or simulation of the network, it eventually gives the scores to each node of 164 protein. Meaning after this, we can just using Excel and then paste the gene name, protein name, and then scores. And then just by clicking, we can sorting out, meaning we can immediately know among those 164 what are the most important, second important, third important genes. So this is very powerful. I'm drug hunter, and I'm not the only one using this network algorithm. Netflix or TVing or TVN, I think in Korea, it often recommend another new drama, new movie. You may like to watch this one. How they do? They're using movie drama as a node like this, and then connect the node by same genre or same director, same actor, actress. So then if you didn't watch and they run the algorithm, and then they recommend highest kind of ranked drama. In LinkedIn, Facebook, Twitter, Blue Sky, whatever, it consistently asks you to connect new people. It's exactly the same principle. Amazon in Korea, Coupang, I think it does the same. They always try to sell more product for you. So they recommend a new product, maybe you like. So this is the same principle. In our case, I think to me it's even more exciting because we are using this tool to identify our lovely protein genes that are causing the problems. And then we may have an opportunity to make a drug. So this is kind of network analysis. And we used this type of approach to many different areas like cancer, Alzheimer, diabetes, and obesity, and so many different stuff. However, this is just an example like in 2020 when COVID outbreak comes to us, we apply this approach to identify some drug for COVID-19. And in this work, we eventually published in Science Advances, but what we did was using the same principle to identify totaling, there was nearly 2,000 approved drugs in FDA, and then we identify among those 2,000 only 200 promising targets, and then further down we did more studies, and finally we tested 5 top predictions on the bench in 2 different wet labs and 2 different cell lines, and we finally identified 2 of 5 really have the kind of effect on the inhibition of SARS-CoV-2 regeneration. So this is one work we did. I'm showing this is like we are now having another, hopefully we don't have any other outbreak, but we are suffering from some kind of, not everyone, but from long COVID, which is, as you can see here, I don't have time to explain, but it's much, much more complicated kind of pathology, and quite a lot of things are connected. But actually, so far, not a lot of drug has been developed, or not many companies pursue to make a drug. So we wanted to apply the quantum work to this, and also we wanted to compare our result with the classical network algorithm. So we call this is quantum-based network analysis, and thanks a lot to Professor Josa. He already gave fantastic kind of background about the quantum, so I don't really need to do it. So thanks for that. But first of all, we need to understand the random walk. As I said, this is invented by Alan Turing, and then basically there is a worker, and then the worker, as you see in the name, this worker just randomly walks through whole networks. And this worker is not really the human, so this guy is not tired by doing this, so it did like 1,000 million times, and then eventually it record in the node, like this path I've been how many times, something like that, and then eventually that calculate the probability, and this is eventually come to the importance of the node in the end. But in quantum situation, as Prof. Josa said, quantum, one of the main characteristics is superposition. So in using by this, the worker is not just walking one path, but the worker can walk through 2 or 3 or 4 different paths simultaneously. So this is somewhat really breakthrough, and then what that means is like it could give us somewhat novel insight. And then I'll come back to this, how we find this. So this work has done by Jessup and Woo-Chang, two fantastic Korean postdocs, and we use the same principle as I show in drug discovery, and we constructed this SARS-CoV-2 in this kind of protein-protein interaction network. And also another reason we're choosing long COVID as an example disease is it has obvious starting node, so SARS-CoV-2. So SARS-CoV-2 comes to our body, the virus, and then it contacts to ACE2, I think you heard a lot of names, and then eventually that causing the problem. So as you can see, SARS-CoV-2 is here, and then we defined 2 different nodes. Like one is a shallow node, which is closer to SARS-CoV-2. Deep node is a little bit further down than the shallow node, means it's far away from SARS-CoV-2 in the data structure and the graph. So we wanted to just like by eye, we saw it, but we loved the numbers. So we just made a quick histogram. And indeed, the majority of the protein are in the kind of in between the second and third kind of distance. Means some of them are in shallow node, some of them are in the deep node. And then also we wanted to check this type of the blue dots, where they are more or less. And as you can see, the depth is more or less in the second and third, but in compared to the old node, I think there is more enrichment in the depth in the deep node. So depth is like the proteins that are changing their expression pattern because of the infection, and the blue one, the deep node, is a directly interacting protein to SARS-CoV-2, so it completely makes sense. This deep node is closer to SARS-CoV-2, and the depth node is a little bit far away. So this is kind of the then ground truth. So we found the long COVID biomarkers from the literature and the clinical trials, and then we mapped where they're actually based. And as you can see in the blue dots, they are somewhat in first, but mostly in the second and third kind of the layer. And we ran the quantum-based network analysis and also conventional classical network analysis. And this is the result we found. By the quantum and the kind of classical one, I think the number of nodes they identify as a key node is quite more or less similar. However, you can see the distribution is quite different. And the quantum-based one indeed identifies somewhat the more or less important key protein in the dim node. But as you can see in the classical one, it's tended to identify the closer to the starting node and not really in the dim node. So this is one of the kind of interesting finding in this work. And then also we wanted to then check what kind of pathway or biological mechanism is being involved. So this one is in the random walk result, and this is a quantum-based random walk. And on the right-hand side on your screen, is a shallow and deep node, and then these colors are more or less random walk identified, quantum walk identified. And obviously, in the quantum walk, it did have quite a lot of interaction from the deep node. In contrast, the random walk doesn't have a lot. And then more importantly, the functional ones, I think this is, maybe you can see here, there's a quite bluish kind of colors from a number of kind of biological pathways. But in here, in the quantum walk, one of the which is remarkable one is there is more neural related pathways that come up. So we had kind of results for that, and then we reported in the paper. And due to time, I'm just going fast, but this is another kind of interesting study about top 10 protein from this quantum walk-based results. And as you can see, there are 10 proteins, and interestingly, only key cancer hallmark gene TP53 are kind of more or less same probability between the quantum-based and classical random walk, but the others are really far ahead of this random walk than quantum. So we identified those 10, and among those 10, maybe you already noticed, but 3 are more or less have a similar name of the protein. So CISD1 to 3 are the family proteins, and this is quite well-known protein for iron sulfur binding domains, and their role is quite interesting as it shares this type of typical domain among those 3 proteins, and this is well known already in the long COVID kind of association, particularly with ROS metabolism. Means it's very important for controlling this metabolic process in the body when we have the COVID. So this is one of the part of the result. And the other one is dive into a little bit. This is a last slide. And CISD1 to 3, and also other kind of related genes. They are all from our network. And then we just focused on this. And we also see that these are really enriched in the mitochondrial and also ER region. And then this regulating of this ROS is also related to calcium ion channels, and this eventually having some problem of the dysfunction of the mitochondria, which is also, again, very well-known factor for the long COVID mechanism. And one of the last and very important thing is we identified the protein called VDAC1, which is maybe promising target for long COVID. And we searched, and there is already kind of developed drug for this particular protein, and this one is also been tested in the long COVID symptoms. So we kind of double-checked whether our findings in drug discovery situation is also working or not by using quantum, and I think we kind of show there is a kind of promise that maybe quantum bring in some kind of novel insight than conventional AI tools. So in summary, we applied quantum, and it offers a deeper exploration of the global structure of the really big network. And nowadays we have more biomedical data, so the network is getting bigger. So the quantum could be very important for this. And also quantum work enables more precise identification. As Prof. Zhang just said, it's very important to having a more accurate prediction. So this, I think, somewhat we validate this hypothesis. And lastly, it's actually uncovering unseen critical disease pathways and also identifying novel therapeutic targets. So there is quite a lot of potential, I think, in our results. So thanks for entire my team and the funders and most of the collaborators. And I will stop here. Thank you.